In this video, we're going to talk about fatty acids and triacylglycerol. And I want you to be able to answer the following questions when you're done watching this video. I want you to be able to compare and contrast saturated fatty acids from unsaturated fatty acids. I want you to be able to answer the question, what is the difference between cis and trans double bonds? I want you to be able to name a fatty acid using proper nomenclature. How does the structure of a fatty acid affect its chemical properties? For example, the melting point. What components are necessary to make a triacylglycerol? Why can cells get more energy from fats than carbohydrates? And what reactions are necessary to begin oxidizing triacylglycerols? Here we have two fatty acids, and what you notice about each of them are they are very simple. It is a carboxyl group and then a long carbon chain. A carboxyl group and a long carbon chain. Now that carbon chain can be between 4 and 36 carbons long. And sometimes there is not a single carbon-carbon double bond between those carbons, and so we call that a saturated fatty acid. It is therefore saturated with the most hydrogens that it can be, that those carbons can be bound to. Whereas an unsaturated fatty acid has at least one carbon-carbon double bond, which means each of those carbons are bound to one less hydrogen than they would normally be bound to, and so this is unsaturated with hydrogens. So that's the sort of the way you can remember the difference between saturated fatty acids and unsaturated fatty acids. The existence of carbon-carbon double bonds means that there are less hydrogens bound to that fatty acid. Now this particular double bond is a cis double bond, and it gives that important kink to that fatty acid, and we'll come back to that in a minute. Now, to name a fatty acid, the first thing that you do is you count the number of carbons. So here we have 18 carbons. And then you count the number of double bonds. We have one double bond, so 18:1. And then in parentheses, you then determine, you look for the double bonds, if there are any, and you count, starting from the carboxyl group, the first carbon of that carbon-carbon double bond. So 1, 2, 3, 4, 5, 6, 7, 8, 9, and so then in parentheses that would be delta 9. So 18:1 delta 9 means we have an 18 carbon fatty acid, one double bond, starting at carbon number 9 when you look, when you count from the carboxyl group. The physical properties of fatty acids depend upon whether they are saturated or unsaturated. Saturated fatty acids are linear, and therefore they stack on top of each other very well, and therefore will be solid at room temperature, so they have the higher melting point. The unsaturated fatty acids are going to have one or more double bonds, and meaning they're going to have one or more kinks in them. They cannot pack very closely together. They don't pack well or interact well with one another, and so they will have a much lower melting point and be liquid at room temperature. In addition, the longer the carbon chain is, the higher the melting point will be, because there is more availability, more space that they can interact with one another. And the shorter those carbon tails are, the lower the melting point will be. Now, most, if not all, double bonds within nature, so all fatty acids that are unsaturated in nature, are going to have these cis double bonds. And cis means same. And what that means is the two hydrogens here connected to these two carbons are on the same side. And that is a type of double bond that gives a kink to it. We have enzymes that recognize that and then can oxidize and digest that particular type of fatty acid. However, there is a process called hydrogenation that companies use to convert unsaturated fatty acids to, or try to convert them to saturated fatty acids But what that ends up doing instead is it changes the cis double bond into a trans double bond, where the hydrogens now are on opposite side of that double bond, and notice there's no longer a kink. So trans fatty acids have trans double bonds. They are technically a unsaturated fatty acid, and therefore you would think, okay, hey, it's an unsaturated fatty acid. Those are the good kinds of fatty acids. Awesome, let's eat lots of that. And the problem is, because it's a trans double bond, even though it's technically unsaturated, it is linear and therefore chemically will act as if it is a saturated fatty acid. And so, and we don't have enzymes to deal with that trans double bond. So a while ago we figured that out, and so now manufacturers are required to tell us how much trans fat are in their products, and it turns out that trans fats aren't very good for us, and so little by little by little we've been eliminating them from the various goods that we eat. And there in fact are cities and countries that have completely banned and outlawed trans fats. Okay, so how do we take our fatty acids and how do we use them to make triacylglycerols? And the answer is we need, as the name suggests, a glycerol molecule. A glycerol molecule is 1, 2, 3 carbon chain, each one of those carbons bound to a hydroxyl group. And to each of those hydroxyl group, via an ester linkage, we will bind 1, 2, and 3 fatty acids. And that's an ester linkage. The carbon from the glycerol molecule to the oxygen here to the what used to be a carboxyl group. And so this is called an ester linkage. Now the 3 fatty acids attached to that single glycerol molecule can be the same, they can be different, as you see here. And we say now that this glycerol molecule has been acylated. So this fatty acid is now considered an acyl group when it is connected to a glycerol molecule. If you take a careful look at a fatty acid and compare it to glycogen, and you compare the reduction state or the oxidation state of the two, you will see that the typical carbon in fatty acids is much more reduced than the typical carbon that you would see in glycogen, for example. So the typical carbon in a fatty acid is connected to 2 hydrogens and then 2 other carbons. Whereas a typical carbon in a glycogen is, so if let's take a look at this carbon right here, is connected to an oxygen, is connected to another carbon and a hydrogen. Okay, so all of these carbons are connected to at least 1 oxygen. So much more oxidized state than what you see the typical carbons in the fatty acids. And therefore, fats provide 6 times the metabolic energy than glycogen. They simply start out at a more reduced state, which means there are more electrons, more hydrogens that can be harvested from them and used in the citric acid, in the electron transport chain. Now, if we are looking at the oxidation of a fat, of a triacylglycerol, the first step is a step catalyzed by lipase, which hydrolyzes the 3 fatty acids away from and off the glycerol molecule. Now, those 3 fatty acids are going to go and be involved in the mitochondria in what's called beta oxidation, where they are clipped. Every 2 carbons are clipped off as acetyl-CoA, and we'll look at that at the next video. Now, glycerol, however, will be involved in reactions that you've already seen when we talked about gluconeogenesis into dihydroxyacetone phosphate. That can either be involved in glycolysis to be converted to pyruvate, or it could be involved in gluconeogenesis to be used to make glucose. All right. So now we're all set to understand fatty acid oxidation, and then eventually we're going to look at how to make a fatty acid. So make sure that you can address each one of these questions from this video. Watch it a few times and make sure you can answer this stuff.